You're very welcome to put questions in the chat as we go along, and we'll have questions at the end, so you can either ask them then in person, or we'll read them from the chat. And just to say that the next webinar, I don't have the exact details yet, but it will be Professor Felix Wackers from BioBest, who also has associations with University of Lancaster, and he'll be talking about biological control outdoors with natural enemies, and that's on the 13th of April at the same time. So now we'll move on to today's topic, which is about phage therapy in agriculture, and I'm going to hand over to my colleagues Mojgan and Shannon, and they will be talking you through the topic and explaining what it's all about. And I'm just going to hopefully stop sharing my screen. Lovely. Right. Okay. Over to you two. Thank you.
Perfect. Just let me share the screen and the presentation. Okay. Could you all see the screen? Could you see my presentation?
Yeah. Yes. Lovely. Yes.
Okay,
thanks, Mojgan.
great.
Yeah. Okay. Thank you.
All right. Hello, everyone. Thanks for joining. I'm Mojgan Rabiey, Assistant Professor at University of Warwick, and I'm joined by my colleague and team member, Dr. Shannon Greer, and we are going to talk about how we use phage to control important crop diseases. And so I'm going to tell you briefly about the crops that we're targeting, the pathogens we're working, and how we use phage to treat those pathogens. Right, so in the UK, did you know that we produce around 300,000 tons of vegetable brassicas that is worth around £400 million? So you could see that vegetable brassicas, including kale, cabbage, cauliflower, almost anything green that we consume every day is part of this vegetable brassicas. We also work on cherries, and cherries worth around £50 million to the UK economy, and UK produces around 12,000 tons of this. So you could see how important these crops are for UK agriculture and economies. Also, in terms of global production, it is very important. So you could see around £52 billion worth of vegetable brassicas and cherries are produced each year. So in terms of global production, again, important for agriculture and economies. So, but over a third of these global crop yields are lost each year because of pests and pathogens, and this results in billions of pounds of annual loss. However, with climate change, we have the spread of new pests and pathogens, but also a spread of existing pests and pathogens. So we have many outbreaks, especially with extreme weathers like extreme rain, extreme flood, extreme heat and drought. So this going to impact the agriculture productivity. So this map here shows the change in agriculture productivity from between 2003 and 2080. So you could see that the darker the red color, the more products are being lost due to climate change. And this is in combination with this crop pests and pathogens. We will see many crop losses, especially as the global population is increasing fast. We need to increase our food production by 70% by 2050 to be able to feed the global population. So it's more than ever important that we understand how we could manage and control our crop diseases. So one of the pathogens that we work on is the Xanthomonas campestri. So it's a bacterial pathogen causing black rot disease in vegetable brassicas. So University of Warwick, in collaboration with Exeter University as part of Xanthomonas Threat Consortium, sequenced around 500 of these XCC isolates. And they found that these XCCs are quite genetically diverse. There are at least more than 9 races of this around, and each race has their own pathogenicity and virulence. Therefore, this is a very complex disease. So this phylogenetic here, you could see that race 1 in dark green and race 4 in purple, they are the most prevalent races identified. So XCC infects leaves and vascular tissues. So you could see these V-shaped symptoms on the leaves of different vegetable brassicas, but also it infects stems and heads and leading to soft rot, stunt growth, but also complete crop failure. So yield losses of up to 50% has been reported in infected fields, and currently there's no control measures. So the traditional method of using copper is limited because of bacterial resistance emerging, because of adverse environmental impact, but also because of the legislations. Also, it's quite hard to breed for resistance, considering that there are many different races and also many different strains within the same race with different pathogenicity and virulence. So farmers usually need to remove the infected plants one by one. So in a huge farm, this is quite impossible. It's quite time consuming, expensive, labor intensive. So farmers might need to abandon their field. That's quite devastating for them in terms of the production. So another disease that we're working on is bacterial canker of cherry, another complex disease, and that is caused by this bacterial pathogen called Pseudomonas stringi. There are many different pathovirus of Pseudomonas stringi cause cherry canker. So we have Pseudomonas stringi, pathovirus stringi, and pathovirus morusplenorum, and then we have different races. So from now on, I call them PSS and PSM, and this phylogenetic is from our recent sampling from across the UK in collaboration with NIAB. We isolated many Pseudomonas stringi from cherry orchards, and we sequenced this. So you could see there are 13 different phylo groups, and with PSS and PSM race 1 and race 2 belong to different phylo groups, and each phylo group, they have their own pathogenicity and virulence. So these pathogens could infect all parts of the tree. So you could see them making shot hole on the leaves. And blossom infection is the most critical and important stage because it leads to necrosis on the fruit. And then we have huge amount of oozing and bleeding from the trunk of the tree that it seems trees bleed to death. So these pathogens is around all year round. As I explained, it could infect all parts of the trees. So yield losses of up to 30% has been reported in infected orchards, and currently, again, no effective control measures. And we can't use coppers, as I explained. We can't breed for resistance, again, because of the complex nature of the disease. So farmers have to chop down the trees, infected trees. So there are 3 to 5 years lost productivity after replanting. So then again, this is quite devastating for the farmers. So there's with no current control measures, as I said, there's no targeted bacterial control, no targeted blossom treatments, and no sustainable biological solutions. So therefore, we need there is an urgent need for fundamental alternative approaches. And one of those approaches includes using bacteriophage or phage or naturally occurring viruses that infect and kill bacteria. So these phages are the most abundant microorganism on Earth, and they are basically everywhere. So you could find them in water, soil, leaf, bark, in human guts. So phages are absolutely everywhere. So how do they infect their host? So they bind to the bacterial cell surface, or we call receptors, and they inject their genomic material inside. They take over the replication machinery of the bacterial cells. They make their own particles, and they lyse and burst the cells open, and each phage goes and infect the neighboring cells. And that's the beauty of using phages, because one bacteriophage infects one bacterial cells, at the same time it produces hundreds of other phages, and they could be used to infect others. So phages are highly specific, and so it means that they only infect the bacterial host they come from, so they have no impact on other beneficial microbes. So they are self-amplifying. It means that they multiply exactly where infection occurs, and they are compatible with integrated pest management. And therefore, we are using the phages to treat cherry canker and black rot disease in brassicas. So for the first part of my presentation, I'm going to tell you a little bit about our progress on black rot disease and phage system. So we went to different brassica farms and we collected samples from soil and leaf, and we isolated many phages. So for example, you could see these plaques in the agar plate. So these plaques contain. So if you could mute yourself, that would be great. Right, so you could see these plaques on the agar plate, and these plaques contains millions of phages, and different sizes and different shapes appears to be different phages as well. So what we do then with these phages, we purify these phages, and then we do a lot of characterization in the lab. So one of the characterization we do genome sequencing. So genome sequencing is to show how safe phages are, so they don't carry any virulence genes, any antimicrobial resistant genes, and they are purely lytic phages. It means that they could just burst and lyse the cells open, and they are used for biocontrol. Then after we know that phages are safe, we do further characterization, including transmission electron microscopy. So you could see this phage, the head, and has a long tail belonging to cyphoviruses. So we do, as part of our characterization, we do a lot of killing curve assay or plate assay, where we measure optical density or population density of bacteria over time. So you could see here, and over 24 hours period. So you could see the XCC in blue line as on its own. So the pathogen grow exponentially during the experiment. However, when we add phages to XCC, they reduce the bacterial population immediately and significantly, and they keep the population down during this 40 hours period. So we know that these phages are very effective in killing and reducing the bacterial population. So here, we then want to understand how broad is the host range of our phages. We test them against many different XCCS strains. As I said, at Warwick, we are very lucky to have a library of these XCCs. So here you could see there's 290 XCC isolates. So apologies for this being a small font size, but just to fit them all in. So 219 XCC isolate here. And we tested 3 of these phages against this panel. So the darker the green means the phages could fully lyse the XCC, and the lighter the green means they semi-lyse, and white means that they cannot lyse. But you can see these 3 phages could kill 50% of these 219 isolates. And they are specific to race 1 and 4, and these are the most prevalent races in the UK. And our screening also shows these phages don't kill beneficial bacteria, such as Pseudomonas fluorescens and SWAM. So we are testing more phages against the 500 XCC isolates. So this is for us to understand how broad the host range of each phage is, and then we could use them for further analysis. So if I put this in around the phylogenery that I showed you earlier, I put these dots around so it's easier for you to look. But so you could see that in the outline layers, if gray means that we haven't tested yet, but for those phages that we tested, you could see that they could kill most of the race 1 isolates and race 4 isolates. So they're effective in that term. So it's easier to look around the trees and see what phage could kill what strain. Okay, so for us to use phage in against different XCC isolates, so we want to design a phage cocktail or phage mixture, and that contains many phages that they target different races, but also target different isolates within the same race. As I said, so XCC is quite complex with many different races and many different genetic diversity. So one of the factors that we need to consider for this is the phage receptors. So in my video, I showed you that phages bind to the bacterial cell surface, or we call receptors. So bacteria and this bacteriophage could use many different receptors on the bacterial cell surface. So this is a gram-negative bacteria, and in the outer membrane, and they have flagella, they have pili, porin, they have lipopolysaccharide, they have capsule polysaccharide, exopolysaccharide, and bacteriophages could use any of these as the receptors to bind to the bacterial cell surface and start the infection. So for us to design a phage cocktail, we want to use phages that they target different receptors, and therefore is easier to target many different races, but also different strains and isolate within the same race. So we did a series of artificial resistant mechanism experiments in the lab, and we genome sequenced that bacteria after, and we found, for example, this phage 1.5 uses lipopolysaccharide as its receptor, and phage 2.2 uses flagella as its receptor. So you could see that this phage is quite ideal for us to use in a phage cocktail, because not only they don't target the same receptor, they don't antagonize and compete with each other, and they're as good to, and they target different races. Another factor that we need to consider during our our cocktail design is to understand our anti-phage defense system. So bacteria also has systems that they could defend themselves against the phages, and we call them anti-phage defense systems. So you might have heard of CRISPR-Cas system, abortive infection, where bacteria do the suicide to kill themselves, so not to get infected, and many different systems. However, phages also evolve around this anti-phage defense systems and are still able to infect the bacterial cells. But for us, we wanted to understand like how diverse this anti-phage defense system is within the bacterial genome. So again, 219 XCC names here. And then you could see the anti-phage defense system mechanisms here, and then the phage host range in green. So the dark blue means the anti-phage defense system is present, and white means this is absent. So here, if I focus in these these panels here, so for example, when phages could infect this bacterial isolate, this anti-phage defense system are lacking, and when there's phages cannot infect, there are many different anti-phage defense systems present. However, these systems are quite complex, so we haven't found any correlation yet between anti-phage defense system and the host range of phages. So this is the thing that we are still testing, and we are going through all these 500 XCC genomes to understand this anti-phage defense system in details. So, next stage, as I said, we want to make a phage cocktail or mixture, and that we could use for our system. So here, again, we did a killing curve assay. So we have XCC on its own, we have XCC and individual phages, and we have combination of 2 phages and then 3 phages. And you can see that with a phage individually and in a mixture, they immediately and significantly reduce bacterial population over time. So that's quite significant to see that all phages work nicely together, but also reduce the bacterial population significantly over the period of time that we tested. So for our implant assay, so we do a lot of implant assay. We are still developing this idea further. But for example, in this case, you can see we have the brassica leaves where we have pathogen only, and we have pathogen and phage treatment. So 14 days post-infection, you could see the XCC spread all around the leaves and into veins and stem. However, when we have the phage application, 14 days post-infection, you see we hardly see any bacteria or XCC spreading into the leaves. So that's quite promising results. However, we are testing different delivery methods at the moment to understand how best we could deliver phage to control XCC population. So this work is still in progress. However, our results are quite promising so far. So I'm now moving to the cherry canker phage system. So we are very advanced with our cherry system. So I'm going to tell you a little bit about this in details. So for our cherry canker, again, we went to different cherry orchards in the south region in the UK. And we collected many bark, soil, leaf samples, and we isolated many phages against PSS and PSM. And you can see the transmission electron microscopy again. So we did a series of characterizations. We have done the genome sequencing that we know that these phages are safe, lytic phages, and they belong to different phylo groups, with many of them being unique and reported for the first time. And from each group, therefore, we chose one phage, and the rest of the presentation focuses on these 5 phages. Again, so we do a lot of killing curves assay, and that, for example, you could see here the MR6 reduces the bacterial population during the 24 hours significantly. Again, at Warwick, we are very lucky to have access to PSS and PSM isolates. So here you could see a panel of 100 PSS and PSMs, and you could see these 5 MR phages. And these MR phages, again, could kill over 70% of the PSS and PSM isolates, and they don't target any beneficial microbes such as Pseudomonas fluorescens, Pseudomonas putida, and so on. So they are quite safe in that terms, and they have different host range. So these MR phages, they target lipopolysaccharide as their key receptors. So in lipopolysaccharide, in the outer membrane of gram-negative bacteria, it has 3 different sections. So we have lipid A, we have core oligosaccharide, and we have O antigen. So these MR phages, they target different sections of these lipopolysaccharides. For example, they target the core oligosaccharide and O antigens. So how do we prove this? We do a lot of gene deletions. So, for example, this glycosyl transferase family 1 is within the lipopolysaccharide gene cluster, and you can see it's within the core oligosaccharide. So we deleted this gene and we repeated our killing curve assay. So you can see that again, we have the PSS wild type, we have the mutant, we have the PSS and wild type MR6, and how MR6 cannot kill the mutant when we deleted these lipopolysaccharides within. So in total, we deleted 10 genes, and only those genes that we identified during our genome sequencing that they were within lipopolysaccharide regions, we found that they stop the phage infection. So these phages, they use lipopolysaccharide for their infection. Again, so we go through the antiphage defense system. However, for PSS and PSM, antiphage defense system are really diverse compared to XCC. So you can see the panel here is on the x-axis. So these are all the antiphage systems we have identified. And again, you can see the host range here and the panel of hundreds of PSS and PSMs. Although that each PSS and PSM race 1 and race 2, they have their own anti-phage defense system, we haven't found any, again, correlation between the presence of anti-phage defense system and phage host range. However, we are still investigating this further, and so to identify how we could just look at the genomes, identify anti-phage defense system, therefore we could say what phage we could use against each strains of PSS and PSMs. So we made a cocktail of 5 phages then, and we did a killing curve assay. So you could see we have PSS on the blue line here. It grows exponentially on its own. However, when we add each phage individually or a mixture of 5, you could see that it still reduced the bacterial population significantly during the 24 hours period. So again, it shows that these 5 phages work nicely together because they target different receptors, and therefore they could kill the bacterial population significantly. So our implanter assay, also we do a lot of leaf infections and woody tissues infections. So here is the woody tissues that we infected with PSS or with PSS and each phage individually, or cocktail of 5, a mixture of 5 of them. Then we score the disease and we'll do bacterial re-isolations. And if you look at the symptoms, you could see these browning lesions of PSS. MR6 reduced the population, have the symptoms. When we have the cocktail of 5 phages, almost clear the symptoms compared to negative. Also, if you look at the bacterial population count, we have all of the phage treatments reduce the bacterial population significantly. So with this in mind, we have done all these experiments in the glass house where we, or in the lab, when our conditions favor the pathogens, and we have the temperatures, we have humidity and everything that we control. However, when we go to the field, everything is different. So the temperature, humidity, rain, wind, microbial community, and everything is different. So we did the first ever UK phage trial in a cherry orchard, when we had a list of questions that we wanted to understand. For example, could MR phages survive in the field? If they survive, for how long? Would phage resistant emerge in the bacterial population? Does phage impact microbial community, and so on and so forth? So in the cherry orchards, we selected our trees, and then we collected leaf and shoot samples of the baseline samples, and we tested for the presence of native PSS and native phage. When we showed that there were no phages and PSS, we started our experiment that we sprayed leaves and shoot with PSS first, and then after the population established, we sprayed them with phage. And from these samples, we started collecting leaf and shoot samples every day for 4 days, and then at day 30. From all samples, we looked for phage and bacterial population to make sure there's no cross-contamination or there is no movement of PSS and phages around. So our treatments included buffer, phage, PSS, and PSS and phage. We had 20 shoots per treatment and 5 shoots per time point, and we collected samples at different time points. So what I'm showing you here is just the plaque forming unit of phage count. So you could see at baseline, we don't have any native phages against PSS. And then from here, we started the experiment when we applied PSS and phage, and we have different treatments. When we have phage on its own after day 3, you can see that we can't detect the phage anymore. However, when we have the pathogen, you can see that PSS, and we could detect the phages. So because the phages rely on their pathogen for replication, and in terms of safety is very important, because then farmers usually ask what happens to the phages when we don't have the pathogen around anymore. So we know that if we don't have any pathogen, there's no phage. So to understand if resistant emerged to phage application in the bacteria population, so we re-isolated our PSS that we put in the field, and whether the PSS alone or PSS that we after phage application, and we challenged them again with wild type phages. And these wild type phages could kill the PSS population that we re-isolated from the field. It means that resistance didn't emerge in the population. If we had resistance, the wild type phages shouldn't be able to kill this. So again, it says that the phage application in the field is don't lead to any bacterial resistance emerging. In terms of phage impact on the fitness of bacteria, so these are again, so we tested the growth of PSS that we re-isolated after phage application, whether on its own or after phage challenge. And you can see that all have the same growth rate. And so we don't want to create a bug that can grow faster and become more pathogenic. However, you could see that phage application didn't impact the growth and fitness of the PSS during the course of the experiment. So to understand the impact of phage on microbial community, so we did the 16S and ITS sequencing. So 16S to understand bacterial population and ITS for fungal population from the samples we collected at day 3 and day 30. And when we did the statistical test, we found that time had the most important effect, and phages on their own didn't have any impact on bacteria and fungal community. Having said that, when we apply phage as a biocontrol, we don't want to eradicate the bacterial population and open the niche for other microbes to come in. We want to reduce the population below the threshold that phages can, and the bacteria that doesn't cause any damage, but at the same time is reduce the population. So we usually use a low dose of this phages, so we don't eradicate the population totally. So that was a very successful application for us. In terms of the phage application, for example, we could apply to the lesions. We could apply as a preventative measure, a spray method, irrigation system, or seed coating. However, depending on the disease, the application is different. However, for effective phage therapy to be successful, we need to deliver phage at the right time, at the right place, and in a cost-effective manner. So a producer of commercial fruit crops routinely deploy bumblebees to enhance their pollination and their yield. And these bumblebees worth around 700 million pounds to the UK economy and the UK crop production. So we thought that we could use this existing agricultural practice to deliver our phages to the site of infection, that is the flowers. So now I'm going to hand over to my amazing team member, Dr. Shannon Greer, to explain the bee-mediated delivery of phage to control cherry canker.
Hello, everyone. So I'm just swapping seats with Mojgan. We're in the same office. So yes, my name is Shannon. I'm a postdoc in Mojgan Rabiey's lab. I've been working with Mojgan now for coming up to 2 years. And in that time, I've been involved with several different phage-related projects. But today, I'm going to present to you for the last 5-10 minutes of this presentation my favourite project so far. And this was a proof of concept study that we conducted last year, funded by Caris Animal Health, where we looked to see whether we could use bees to deliver phage to control the cherry canker pathogen PSS. So in this study, we had 4 main research questions. The first was whether the buff-tailed bumblebee, Bombus terrestris audax, which is a European native species and a commercial bumblebee species, whether this bumblebee could transfer phage from hives to flowers. The second was whether these bees could transfer phage from flower to flower. And as well as moving phage, could bees also vector the pathogen? Could they move PSS from flower to flower? Are they enhancing the spread of the disease in cherry crops? And then finally, probably the most important question is, if bees can deliver phage to cherry flowers, are these delivered phages effective at reducing PSS populations? And we published our findings for these research questions recently in Biological Control. So if anyone's interested in the detail, the link's at the bottom of the page. But for today, for the interest of time, I'm just going to focus on the last research question, which is, does bee-delivered phage reduce bacterial populations in cherry flowers? So in this, to answer this question, we have two experimental setups. We work in both artificial flowers and real cherry flowers. So cherries only flower for 2 to 3 weeks of the year. So as you can imagine, trying to fit lots of experiments into those weeks is a manic time. It's that time of year again. So we've developed an artificial flower system that allows us to work out of that bloom period. And for our artificial experiments, we have 2 treatments. So we use these mini hives that contain 15 to 20 bees. And in one compartment, we have a clean hive. So bees are released from a clean hive. And in a second compartment, we have bees that are released through a runway where there's phage powder at the entrance. So the bees crawl through phage powder, and then they are released to forage on these artificial cherry flowers, which are printed cherry flowers with a 20% sucrose feed in the centre that we spiked with PSS. And then at given time points, we sample the sucrose feed and we test the sucrose feeds for each of these flowers for the presence and concentration of both phage and bacteria. And we sample the flowers at T0 before bee release, at T1, 1 day after release, T3 and 4, which is 3 or 4 days after bee release, and then T7, which is a week after release. And then we effectively have done the same in real cherry flowers. So again, 2 compartments, a clean hive. The bees are released to forage on real cherry flowers that we've spray inoculated with PSS. And then in the second compartment, we have a hive where the bees crawl through a phage powder runway and they forage on PSS infected flowers. And again, at the same time points, we collect these flowers, incubate them individually and buffer, and then we test this for the presence and concentration of both phage and bacteria. So that's been the setup. A little bit about the results. So in short, in the artificial flowers, bees are very good at transferring phage from the hive to artificial flowers. They transfer to up to 90% of artificial flowers, and the transferred phage was effective at reducing and clearing bacterial infection in these artificial flowers. So this box plot here shows the concentrations, the bacterial concentrations in time. So the higher the CFU, the colony forming unit, the more bacteria. And the red box plots show the bacterial counts for just the control compartment. So it's the clean hive with no phage. And the blue shows bees released, the colony counts for flowers where bees have been through a phage powder. And you can see at T0 before bee release, the bacterial concentrations are the same, which you'd expect. No bees have visited the flowers to deliver phage yet. But even after 1 day, there's a significant reduction in bacterial population, and then we have complete clearance of the bacterial PSS by day 7. So in artificial flowers, we see a similar trend, although it's not so pronounced. We see a reduction in bacterial counts at T1, in particular, and at T7. The bees weren't as efficient as at moving the phage to real flowers. They moved them to about 40% of flowers compared to 80% of flowers in an artificial setting. But a lot of the differences, I think, are down to the experimental setup and design. So the artificial flowers are a optimal environment for both the bacteria and phage. It's a high sucrose feed. And it's only 24 flowers that we sample with the artificial flowers, whereas the cherry flowers, there's many more flowers available to the bees to visit. So what we sample at a given time point may not have been visited by a bee. But we're now working, we've been funded for a further 18 months by Caris Animal Health, who funded this project, to see how we can optimize this in real cherry flowers. So we've been able to show that bees can deliver phage effectively to reduce PSS populations in real and artificial flowers, and it looks like a promising control or dissemination strategy for biocontrol for floral pathogens. What I didn't show was that bees can actually transmit PSS between flowers, but bees that have been treated with phage or crawl through the phage powder no longer transmit PSS, so it breaks that transmission cycle by the bees. And as Mojgan said, these bumblebee hives are used routinely anyway in cherry orchards, so it could be leveraged for targeted delivery of phages. So I'm just going to hand back now to Mojgan to finish the last little bit of the talk.
Right, so why this application and this delivery method is game-changing? So we enable precise, preventative, safe protection at the exact moment and the exact location where the infection begins. And these phages are, as I said, they are very host specific, so they don't target any of bees microbiome, and they are safe for the environment. And then therefore we don't need any spray method, so there is no risk of drifting or waste, or also we have a full coverage as bees visit all the flowers. But also this is not just the single crop solutions, so this model extends to many more other crops. And the UK opportunity alone for cherry is exceed over £100 million worth in terms of potential economic saving with far greater global scalability. So in that note, I want to thank everyone involved in the projects and all the funding bodies, and thanks to my team, and thank you all for listening, and I'm happy if you are happy to answer any questions.
Excellent. Thank you both very much. That was really interesting. So we've got a few questions in the chat. Can you see them, Mojgan? Or
No, I can't see the chat.
no? Okay. So the first question was, what is OD 600? But Dave Chandler answered that in the chat, and I think probably pretty comprehensively, in case unless anybody wants to challenge that. So we'll go on to the next one, which is, can modified bacteriophages be considered as GMOs?
Right, so yes, but we are not using any modified bacteriophages. So many systems, for example, in animal and human health, when you use phage and for clinical settings, they might use modified phages. However, for us, we use phages that they are natural, so they are naturally occurring viruses that we collect from the environment. We don't do any modification to them, so they have. In that terms, they are safe, but in terms of application, yes, in other systems, these modified versions are being used. For example, like in other systems, they try to, for example, increase the host range of phages by modifying the genomes. But for us, we want just to use natural phages with no modifications. And so in terms of the use, they are safe to be used. Yeah, so they're not modified at all.
Great, thank you. So the next one, and this was a graph quite early on. What will happen to your graph if you keep the phage and the bacteria for more than 24 hours? And I can't remember which graph it was.
Yeah, no, I understand. So yeah, so we usually in publication and stuff, you see that they only show like few hours of the experiments. So however, we show 24 hours, but we keep these experiments for long. So we usually do 72 to 96 hours of experiments. Still phages significantly reduce the bacteria populations throughout the time. And so that was in vitro assays. in plant assays, we keep them even for longer. So we do weeks of experiments and we collect samples at different time points. Again, we see that phages reduce population throughout the experiments.
Great. Next one is there any scope for this to be developed against fungal pathogens?
Yes, so there are phages that they could target fungal pathogens. However, so far we only focused on bacterial pathogens, but we are also interested in phages that infect fungal pathogens such as Fusarium. So that's our interest as well, to find phages against those.
Excellent. Now we're changing crop. What are the practical applications and breakthroughs of phage therapy on bacterial black spot of mangoes?
Oh, mangoes. Well, so phages could be used against any bacterial pathogens. As long as we find the phage that could infect the bacterial host, we could develop this further. So in terms of mangoes, if the flowering stage, for example, is important, then again, we could use the delivery system that we're developing at the moment with bees, that could be used. But however, again, is a matter of finding phages against the bacterial pathogens of interest. So, for example, in this terms, it's a black spot in mangoes that you mentioned. As long as you have the phages, we could apply them against the pathogen. So far we have a library of phages that could infect Pseudomonas stringis. We have phages that could infect Pseudomonas savastinoi that infect the ash, causing ash canker. We have phages that infect pathogen causing acute oak decline. And we have phages that infect Xanthomonas campestri. So we have a library of phages that target many different bacterial pathogens. So it's just a matter of like what the interests are and where we can find the phages, and then we could develop them further for application.
Great, thank you. Are there any regulatory hurdles for phage therapy in agriculture?
So this is the thing we are trying to understand. So how we could get this phage to the market? What are the regulatory framework and how long it's going to take? So this is in progress. But the hurdle is like again same as any other biocontrol product that we use. We need to understand the safety. So has safety are for the environment, has safety are for the fruit production, has safety for the consumption and so on. So we try to understand. The more research we do, the more we understand about the safety, the application methods, and then we could get through the regulatory frameworks, and then we try to get that to the market.
Great, thank you. Next one is, are there any implications for the bees of carrying phages?
So yeah, that's something that we're working on this with this current funding, this follow-on funding we've got from CARIS. So we're doing some microbiome studies to see how the phage affects the microbiome of the bee and the health of the bee. But because they're a commercial commodity, they're like a seasonal disposable commodity. So yeah, so we are working on that. We're doing some microbiome studies. Yeah.
Great, thank you. Right, changing crop again. What are the prospects for phage therapy in the fight against the bacterial burn Erwinia on apples?
Yes, so this is another system that we have started working on. So we currently started working on using phage to target Erwinia amblivora that cause fire blight in apple and pear. Again, so we are testing the system at the moment. We are trying to understand the phage host range and so on. But also as for apple and pear, same for cherry, the flowers are the most important stage. So this is a delivery system that we already developed. So we could also test that against fire blight of apple and pear and see how we could target Erwinia and fire blight.
Great, thank you. Next one is, could you use phage to target the bacterial endosymbionts of insect pests?
Oh, that's quite interesting subject. Yeah, so we were thinking about this. So I guess so the difficulty is just to get to that in the symbiosis. So it's within the system. But as I said, if you have the right phage against the right host or the right pathogen, we could use that to treat them. So there's a possibility.
Okay, which the aim being to presumably weaken the insect pests.
Yeah, exactly.
Yeah. Right. Okay. So then there's a comment from Ray Cook, which is that CRD HSE consider the use of phage as a low-risk microbial biopesticide, as regulatory approval is a big potential barrier for phage-based PPPs. How do you consider phage could be de-risked to speed up approvals?
So I think for us is the more data we have to prove the safety, the quicker we could get to that point. So that's why we try be doing for example, we did the first UK field trial. We had the license to do that. Since then we have done another field trials in a forest as part to protect the oak trees. We also with the current bees experiments, we're trying to do polytunnel experiments to understand the phage efficacy, safety, and so on. So the more data we get for the safety measure, the better we could answer all the questions that the regulatory framework might have, and we could quicker get that to the market.
Thank you. Next one is, did your phages reverse your cabbage leaves from necrosis state to healthy state?
Yes, so for the ones that the experiments that I showed you, we did the pricking. So we infected the edge of the leaves with the pathogen. That's where the infection occurs, and where it initiates the infection. And then we apply phages to the same infection method. So so we didn't see any symptoms after we applied the phage to those leaves. And so it shows that it stopped the pathogen from spreading and it stopped the symptoms. However, since then we are doing more experiments. For example, we want to see how best we could apply phage to brassicas, whether it could be through a spray method, it could be through seed coating and so on. So we're doing some trials to see what methods is the best to treat the XCC from spreading and then keep the plants as healthy as possible. So this is an ongoing project for us at the moment.
Great. Right, next one. Has phage treatment been validated as seed treatment for seed-borne bacterial diseases?
So as far as we know, so this is what we're trying to do in terms of XCC infection. So we want to see if we, for example, could do seed coating to stop the pathogen spread. So, for example, in terms of XCC, could also move from seeds to So there's a seed transmitted disease. So we try and get different protocols to see like how best we could develop our seed coating method to deliver phage. So this is, as I said, it's an ongoing project. But yes, we could use this phage as seed coating, but we have to have the right product for the seed coating because it's in the soil environment and it's different from the flower stage or leaf infection.
Great. Next one. Can you tell us if similar research on use of phages for bacterial disease control is happening in other places around the world?
Yes, so in USA, currently there are commercial phages available that they target Pseudomonas stringy, they target Xanthomonas campestris, they target citrus trees. But the regulations is quite different in USA compared to UK, so we don't have it here. We have that also in other European countries. So there are phages product out there in other countries.
Great. Now we're back to seed treatment again. If applied to seed coating, how long can it survive on the seed and what are the conditions that seeds need to be stored at before they could be used?
Yeah, so this is, as I said, this is an ongoing project. So we're trying a different formula to see how best we could make our seed coating with phage. Not only phages could survive the longest, but also they keep their viability, and therefore they could help seeds to don't get any infection from pathogens. And in terms of storage, again, we need to see how we could increase the shelf life of not only phage, but also seeds that are treated with phages. So this is ongoing projects, but hopefully maybe in the next presentation we have more data to show.
Thank you. Next one, would phage treatments have varying efficacies against different strains of the same bacterial species?
Yes, so I showed you a panel of we tested, for example, phages against a panel of 219 XCC isolates from different races, but also some within the same race. And yes, so phages have different efficacy. Some that could fully lyse and come semi-lyse, some might not be lysing at all. So this is we're trying to understand. So as I said, we're trying to understand, for example, what receptors phages use to bind to the bacteria cell surface, what antiphage defense system are present, how do we know what phage to use in our phage cocktail that we could target many different strains of the same race or different races. So far we've seen that there's no correlation between, for example, the presence of antiphage defense system and the phage host range. And we found that in our recent publications we showed that it's still phage screening is the best method for us to understand the phage host range, which phage could infect which bacterial pathogens, and then we could decide what to use. So again, although it's time consuming to do the phage screening, but it still is the best method rather than just relying on genomics and genome mining.
Great. So bit of a marathon here. So 3 more to go. How much does the crop need to be worth to undertake all the research, development and commercialization to bring a phage to market?
Well, any crop is important. And so you can see that I showed you 2 different examples. So we have cherry that worth around 50 million pounds to the UK economy compared to brassicas that worth 400 million pounds. So both are important for farmers and growers. So we can't put a price on this in terms of farmers, the values for farmers. So we try to, for example, as I mentioned, we try to find phages against other diseases like ash canker, although is not known widely, but it still is around. So we try to see what we could use to target those. Also, it's important, for example, in terms of Irvinia and apple and pear, although it's not widely spread in the UK, it's important we have the right product. So in case there's an outbreak, we are ready for the outbreak so we could treat the pathogen. So any crop is important for us, and we are interested to find phages against the pathogens infecting the crops.
Great. So how stable are these phages?
So we do a lot of as part of our characterization, we try to understand how stable they are. For example, Pseudomonas stringy phages, they are stable. So we tested the stability and viability over a 6-month period, and we know that in different temperatures, from -80 to -20 to room temperature to fridge temperature to 27, and they could survive up to 6 months. But this is a period we tested. So in terms of the shelf life, we know that at least 6 months they are viable.
Great. So we've got one more question and then a comment. So the final question is, will phages have any negative effect on biopesticides that are being applied?
No, so we know that, as I said, the phages are very host specific, and they only target their host pathogens. So they do target, and we know that we use them with other application methods, other integrated. So in terms of combining them with other pest management systems, they are quite safe because they only target the pathogen of interest. They only survive when the host is present, and they don't interfere with any other integrated pest management strategies.
Thank you. And there was an add-on after that, which was about also any side effects on beneficial predators, but probably it's the same response, isn't it?
Exactly. So every time we do our host ingestion, we have a panel of beneficial microbes that we tested. For example, in terms of PSS, we tested them against 30 different Pseudomonas and other beneficial microbes that we know that they are within the cherry microbiome, and we know that none of the phages impact any of those beneficial microbes. They're very host specific.
Okay, so there's a comment from Ruben saying in regulatory terminology, road risk is the lowest possible category, e.g. allows no MRL and longer period of approval. And Wayne has sneaked in a final question, which is what would be the maximum number of phage in a cocktail to maximize the effect of the product against as wide a range of bacteria as possible?
Yes, so as I said, so we in our phage cocktail we try to include phages that they target different receptors, so they don't compete at the same receptors to infection. As long as although we have tested combinations that they use the same receptors and they don't antagonize each other, so we don't want to include lots of phage. So, for example, in the UK, we've done lots of different sampling that we know what the diversity of the PSS population is, or what diversity of XCC population is, and we know that what phages target. So that's why we do a lot of host range assay to understand what if phages have a very diverse host range against XCC strains within the same race or different races. So far we have tested a maximum of 5 phages, and we know that they are very effective. They increase so they target different strains and the different phyla groups, different races. For example, for our XCC phages, we are using 6 phages as a combination. So this is, again, it requires us to do a lot of host range testing to understand the library of phages we have. They are effective against what bacterial isolates, races, phyla groups, and then we could decide how many phages we could include in a phage cocktail.
Great, thank you. Right, so first of all, thank you to everybody who joined us this afternoon, and thank you for a really good long list of questions. And the biggest thank you go to Mojgan and Shannon. Thank you for a really interesting presentation, and to say for fighting your way through all those questions as well. So thank you very much indeed. And thank you all for joining, and hopefully we'll see some of you at the next webinar in April. Thanks a lot.
Thank you. See you later. Bye.